Hello, everyone, and welcome to the Veeda podcast series, where scientists, business leaders, and clinical researchers discuss some of the latest and most interesting drug development topics. Today, we are joined by Dr. Sunil Soitawala, site in charge for our Mehsana Clinical Research Unit, who has an experience of 7 years as a principal investigator, managing a variety of clinical trials and overseeing site management activities. Today, we'll be discussing the key aspects associated with the execution of a recent complex study done at our Mehsana facility of a complex drug called omalizumab. Welcome, Dr. Sunil.
Thank you, Yuvragi. It's pleasure to be here.
So, Dr. Sunil, could you start by telling us first about the drug omalizumab?
Yeah, of course. Omalizumab is a monoclonal antibody that is mostly used to treat allergic asthma and chronic idiopathic urticaria. It largely works by binding to a protein called immunoglobulin E or IgE.
All right. So, Dr. Sunil, I'd like to know about the objective. So, what exactly was the objective of doing this clinical study?
So the sponsor was planning to develop an omalizumab biosimilar and also to obtain the authorization for its global marketing, and hence for that purpose we had to demonstrate that biosimilar drug product is pharmaceutically equivalent and bioequivalent to the reference drug product. So our study mainly aimed to investigate the pharmacokinetics, pharmacodynamics, and immunogenicity of the omalizumab, as well as its safety and tolerability. This study was done in healthy volunteers, which involved maintaining a standard IgE ratio.
Okay, that is interesting to know. So now moving forward, I'd like to know what precise reasons actually led to the selection of Mehsana Clinical Research Unit for conducting this complex BE study.
So, Yuragi, there were three main factors that we considered. The first factor was our state of the art infrastructure that Mehsana Clinical Research Unit has to offer. Our infrastructure at Mehsana includes four clinics with more than 9,000 square feet area, giving us the highest per bed area across all of our facilities. A larger space also provides flexibility and allows us to more easily accommodate changes in the trial design or protocol if necessary, as it has the resources to adapt quickly to the new requirements. The second factor was the screening and the recruitment capabilities of the volunteers. Our clinical research unit at Mehsana is located in an area with over 2.25 million people in the Mehsana district. And this place shares borders with the Ahmedabad in the south and also with Rajasthan state. So location of this place is such that it offers a tremendous opportunity for recruiting healthy and the diverse volunteer populations, which helps us to get the right population for the right study. And the third and the most important factor was our team's experience in handling and executing the complex studies at our Mehsana unit.
Understood. So, Dr. Sunil, since you mentioned in the last point that the team at Mehsana has extensive experience in handling complex studies, would you kindly elaborate on some of these types of studies that the team has handled and the reasons why they are referred to as a complex or a challenging study?
Yes, our team at Mehsana definitely has vast experience in handling a variety of complex studies, such as KCl urine excretion studies, in which samples of the urines are collected and utilized for the PK analysis, iron sucrose, which requires the expertise of the dosing to give the complete drug in the desired period, Aliglustate drug study conducted in the form of the transparent film, which is being the dose sublingually to see the gene effects of the CYP2D6. Various nebulizers and the meter dose inhaler studies in which the drug has to be given by the nebulizer and the meter dose inhaler, and subject training becomes the most crucial part of these studies. Also, we have done propafenone study in which continuous cardiac monitoring along with the presence of intensivists is needed to ensure the safety of the subjects. Therefore, taking into account the complexity of this omalizumab study and our team's expertise and experience of managing a variety of complex studies, this study location was selected at our Mehsana facility.
All right. So, moving on, Dr. Sunil, I am aware that there are a variety of potential challenges that could arise during the clinical study execution phase, particularly those pertaining to operations. So, I'd like to ask you about the key challenges encountered during the execution of this omalizumab study and how the team at Mehsana overcame those challenges.
Mm, From challenges point of view, I believe we mainly had challenges around the screening and recruitment of volunteers, and a few challenges around IMP handling, dosing, ensuring safety of the volunteers, and last but not least, the handling of the PK ambulatory samples. So coming to the first challenge, which was the volunteer screening and the recruitment. So before enrolling the initial research group, we made sure that the study person was chosen from each functional core group, and this was ensured by the entire group leaders and the quality assurance team throughout the study. In addition, the team was continuously in touch with the IGE pass volunteers to call them in for screening as well as to avoid their diversification into the other CRO studies. Our clinical team screening and the recruitment strategies help us to get a total of 1900 plus volunteers with IG screening feat achieved for 600 plus volunteers to get around 358 eligible participants enrolled for this study. So our volunteer recruitment staff has been successful in signing up more volunteers each day for study-specific ICD and the IgE test.
All right. And Dr. Sunil, I think we also do see a lot of pharmacy related challenges while handling such complex studies that can actually impact the dispensing and administration. So, could you tell us if you encountered any such challenges? And if yes, then how were you able to handle or manage them?
Of course, IMP handling was a challenge altogether. And IMP being very sensitive in nature, we had to take extra precautions during the receipt, storage, dispensing, and the transfer to avoid potential damages to the IMP. In addition, the team ensured that at the time of IMP receipt, data loggers were checked by the pharmacist to ensure temperature as per the storage condition, which was maintained a cold chamber with the proper leveling. IMP dispensing was done on the day of the dosing, and for the lyophilized product, the pharmacist ensured reconstitution of the IMP as per the protocol and fill up the syringe according to dose. The pharmacist team ensured that all the necessary precautions were taken while reconstitution. As per reconstitution, usage of an 18 gauge needle, and for subcutaneous dosing, we have to use 1.5 inch 25 gauge needle. So IMP needed to be kept for half an hour to come to the room temperature before dosing as per the study requirement. And if we can see another challenge, let us come to another challenging section that which was related to dosing. And this was challenging in the sense that the omalizumab drug was to be given by the subcutaneous route that can sometimes cause skin injury and the superficial thrombophlebitis if not administered properly. We were able to correctly administer the medication in accordance with the study protocol thanks to the assistance of a skilled team of nurse and the phlebotomist, avoiding any dosing deviations. The clinical dosing staff also made sure that aseptic handling practices were followed. The medical team also monitored volunteers while they were inside the facility, and at each visit for an ambulatory sample for injection site reactions at all post-dose time periods.
Understood. That's very interesting to know. So, Dr. Sunil, next, I'd like to hear from you regarding the volunteer safety measurements that you must have had taken into account during the execution of this study.
For ensuring the safety of the volunteers, the team followed the risk mitigation practices by the maintaining proper communication channels for adverse event assessment, recording, and reporting to the regulatory authorities. There was a dedicated team of intensivists during dosing for up to 6 hours to ensure safety and to handle any unexpected adverse event during the study. Also, the blind nature of the study was maintained by the strong invigilation of the investigators, followed by the quality control and the quality assurance team. The study was conducted in groups as per the protocol. So after each subject in each group was dosed, we had to collect the safety data, ECG, vital, SpO2, safety lab parameters, and adverse events and the serious adverse events data. Also, the investigator needed to seek approval for the subsequent group after presenting this safety data to the DSMB committee as per the protocol requirements before dosing the subsequent group. After getting approval, subsequent groups were enrolled. Other than that, there was continuous monitoring of vitals and ECGs in-house in the presence of experienced cardiologists. Multi-speciality vibrant hospital and an emergency team at the hospital were intimated in well advance before dosing to keep everything ready to mitigate any untoward reactions. The subject had been informed and encouraged to be notified of any discomfort or adverse events after dosing. 24/7 availability of the doctors, nurse around the clock was ensured up to the housing of the subject as per the study requirements.
Okay, understood, Dr. Sunil. Now, moving forward, since earlier you mentioned that IG levels are extremely critical in such types of studies. So, what was the standard maintained for IG levels during this particular study?
Thanks for mentioning this question, Yuvragi. So there were study-specific criteria regarding this immunoglobulin E. So at the time of screening, volunteers' serum immunoglobulin E concentration should be less than 100 international unit per ml as per the study requirement.
All right, okay. So, Dr. Sunil, I would like to know more about how did the team manage a long duration study like this, especially which involved multiple ambulatory samples?
That is a very good question, Ibragi. So we had to identify the subjects from our database before the initiation of this project who had a good history of turning up to the facility for ambulatory samples, considering the long ambulatory samples in this study, and try to get enrolled such volunteers. To track ambulatory, cards were given to each subjects date-wise for the ambulatory samples. Other than this, our team of custodian ensured the sample visit through frequent follow-ups telephonically, and also for a subject who did not turn up for the ambulatory, a home visit was done by the screening team member to make sure that subjects come for the ambulatory without missing the subsequent samples. Investigator also took follow-ups to ensure that all subjects come for every ambulatory visit, and the medical team ensured that all the volunteers underwent each ambulatory visit of the study. Moreover, if subject misses a sample visit due to an unavoidable personal reason, the PI ensured that the subject is contacted as soon as possible by the study staff to make up for the missed visit. Our PIs also ensured that each subject got appropriate medical care promptly from in-house physician and the medical team, which helped us with the better retention of the volunteers.
All right, understood. So, my very next question is actually related to the sample storage and management. How did the team ensure the sample storage and sample management for this study? So, Dr. Sunil, could you give us a little brief on some aspects like, did we have a dedicated team or any specific temperature monitoring processes or systems that ensured a smooth storage and management of samples during this study?
Hmm. A dedicated team of approximately 30 experienced phlebotomists was assigned as per the protocol: sample collection, separation, storage, and segregation. And it was ensured that the samples were kept for half an hour to allow clotting before centrifugation. After the sample centrifugation process, segregation was done by an experienced and skilled team of phlebotomists to avoid any chances of error, followed by the shipment of the samples. I also want to highlight that for sample storage, there was a need of maintaining the cold chain, which was a very big challenge, and that was ensured by the sample management and the bioanalytical team. The cold chain process typically involves the use of specialized packaging and the temperature monitoring device to maintain the required temperature range. So for storage temperature, -70 plus or minus 8 degrees Celsius was the protocol requirement for this study. And the shipment was done in dry ice. Two data loggers for continuous temperature monitoring were placed inside the shipment box along with the sample to ensure that samples were transferred at a controlled temperature and that the integrity of sample is maintained.
All right, understood. So, Dr. Sunil, this will be our last question for this podcast. So, Dr. Sunil, could you tell me what were your key learnings from this study?
Yuvragi, just to mention that while it may seem like a typical BAB study, we have learned that biosimilars present a unique set of challenges at each stage, from protocol to study conduct, relating to complex study design, whether it is voluntary requirement and retention, dispensing, dosing, and all of which really demand specialized skill and the careful planning. Having a skill and the experienced workforce, ensuring the availability of the necessary resources, and providing through training to every team member are crucial components for understanding and achieving the main study findings. And last but not the least, ensuring our volunteer safety was a crucial factor for this research, and our team was trained and prepared to handle any emergencies that might arise. So my biggest learning is to have a plan specific to the nature of the drug being tested and allocation of right resources. These resources might not have experience of working on drugs that are exactly like the drugs being tested, but they must have some exposure to or experience of working in environments that are more like those you might face.
Thank you so much, Dr. Sunil. Uh, Thank you so much for joining us on this episode of our podcast. Your insights and expertise on this particular subject have been extremely helpful in bringing to our attention a number of intriguing facts. And with that, we conclude our discussion on the challenges of conducting an omalizumab study on healthy volunteers. We hope you found this episode informative and gained a deeper understanding of the complexities involved in performing such studies. Thank you for tuning into this episode, and we look forward to bringing you more thought provoking discussions in the future.
Thank you, Yuvragi.